recording has started. Would you like to start?
Sorry, I realized that I accidentally hit my mute button on my speaker. I have started the recording, so I wanted to welcome everybody today, and we are starting a session on experiencing an ANSLO remote web-based meiosis and mitosis lab activity. Our presenters today are Sue Buie and Dan Branan. A little information: we've provided a bio for Sue, and we have also provided a bio for Dan. And I don't want to spend a great deal of time on that because that was also shared on our listserv, and I really want to give each of them an opportunity to really work in relationship with you to show you about the ANSLO lab activities. The last thing I wanted to mention was this webinar is funded by the U.S. Department of Labor, and the funding has been received through a TAA CCCT grant. With that, I'm going to turn it over to Sue and go to her first slide. I hope is the right one. All right, Sue, I'm turning it over to you. Thanks.
Great, thanks, Sue. Well, thanks for having me here. I wanted to talk about the value of remote web-based labs. So we had our first remote web-based lab with human biology A100 students. This is for the occupational endorsement medical coding certificate. So please keep in mind, and I'm going to say this again, that these students are non-science or biology majors. Okay, and so I want to talk about some of the things that we went through, some of the things that we learned. And of course, you can't have a lecture, I find, in academia without using the word collaborative. So I'm really trying to come up with new synonyms like cooperative, concerted, collective, combined, shared, common, take your pick. So I would say that this was definitely a concerted effort between WICHE, Rutgers, NANSLOs, the leads that wrote the original labs, as well as the lab directors, Albert in British Columbia, Dan in Denver. It was, and I know that I'm missing quite a few, Maria Fife. So it's this huge effort to create and deploy this one lab and this system for creating a lab, or a system really, that is going to work best for our students. So the premise that I followed was through Quality Matters. Many of you may be familiar with Quality Matters. If you are, go ahead and give me a smiley face. That's the icon on the top left. Dan and Albert, a couple others, Brenda. I've lost my mouse. Here we go. Julie. Okay, so at least half of the class. So what Quality Matters is an organization that provides quality assurance for online education, and they use a peer-based approach for the continuous improvement in online education and for student learning. So it has some pretty rigorous standards that you need to follow. Okay, it has this rubric. And so in order for your course to be Quality Matters certified, you need to go through these multiple standards, and then a board reviews your course. It takes probably 3 to 6 months to be Quality Matters certified. And they also have courses that they teach for this, and it includes the use of learning objectives, content presentation that aligns with lab activities and assessment. I was particularly interested to take Quality Matters because I think it's important to, especially for us distance educators. So, you know, I have a course that is now Quality Matters certified that gives us that extra, what's the word I'm looking for? Somebody can text it. Well, quality, right? We can provide a quality course even if it's a distance lab, and we have to use multi-modalities to do so. So of course with this lab there were learning objectives, instructional strategies, the content presentation meshed with the lab activities, and then the assessment was that the students were required to provide a lab report, a clear and concise laboratory report using the scientific method. Now, the purpose of this particular lab, which is what I'm going to focus my time on in student response, was mitosis versus meiosis. Using that RWSL lab microscope, which if you haven't had a chance, you'll have a chance to manipulate today. And the purpose was to compare and contrast meiosis and mitosis using that RWSL laboratory. Again, remember, the students registered in this class are pursuing their occupational endorsement and certification in medical coding. They are not, their major is not science-oriented, and all the classes that we have in this particular program now are delivered fully distance. Also keep in mind that this is not a virtual lab. We're going to use the RSL lab. We're actually going to go into the lab through our computer to be able to manipulate a microscope, which you will do on your screen. Okay, the microscope that we use in Alaska is located in British Columbia. The one that you're going to be using today is in Denver, Colorado. The students are expected to collect data by capturing an image and saving it to their computer. This takes anywhere from 45 minutes to 2 hours, depending on the student and the group. Then they're going to take this data that they collected. They're going to need to analyze it, interpret the information, and put it again into a laboratory report. This lab does use multi-modalities: tactile, visual, auditory, clinical applications, and certainly critical thinking, which is key for any individuals that are going, well, into any field, but in particular that healthcare field, right? Use that problem-solving, inquiry-based type of teaching. Emphasis is placed on, as I said, inquiry-based learning. I have a text. Dan said that's 45 minutes to 1 hour to collect several images, not just one. Yes, thank you. So let's talk about this particular lab. Now, here's one of the great things about NANSLO. So they create a lab. Very, very detail-oriented, really quite exceptional. All right? And because it's an open source lab, you as the instructor are able to modify the lab. Now, the lab that they created is fantastic for my anatomy students, but a little bit too difficult conceptually for my non-science-oriented students. So I was able to modify that lab, okay, and make some changes, and add some learning objectives and change some learning objectives. So the learning objectives of this lab was to describe the cell cycle, identify the stages of mitosis. This would include from interphase through cytokinesis, describe the stages of meiosis, define the differences or differentiate between haploid and diploid, display the ability to use a microscope, calculate simple equations, and capture images. And you'll learn more about capture images as you do that yourself. Following the previous 4 objectives then, the students are required to complete a lab report using the information from the previous 4 objectives. They had to gather and interpret that information to form a concise and objective laboratory report, and label then the process of mitosis and meiosis with the captured images and attach them to their report as an Appendix A and an Appendix B. Now, I want you to keep in mind that these students that we're talking about have A, never written a lab report before, or B, haven't written a lab report since high school, okay, which could be 20 to 30 years ago. So there was a lot of instructor preparation, so a lot of information NANSLO had provided me. Some things that from student feedback I felt needed to be added. And so instructor preparation I found is key to the success of the RWSL laboratories. There are several strategies that can be used to make your students more comfortable and prepared for this type of lab. Typically, students are very anxious, especially when trying new technology. I mean, if you think about when we go into even a Blackboard Collaborate session, even if we've done it 100 times, is my mic going to work? Am I going to have problems? Every time you have a presentation, there seems to be a glitch, when maybe you haven't had a glitch for 6 months. So I want the students to focus on the anatomy and physiology of the lab, not the fear of new technology. So some of the ways to prepare for this lab was I went into the lab with Dan and Albert, and we tested the lab out. So today you're going to be sharing Blackboard to get into the lab. I and Susan Merkovich, a chemistry instructor, were able, just like our students, to go right to the lab. Right, a lot quicker response. There's going to be no delay in screen. You're going to see some of that today. And then we also practiced capturing the images, following the instructions of the modified lab to make sure that it was clear and easy to follow. Right. Also met with the lab directors, both Dan and Albert, to talk about the slides that we needed, and the sequence in which we needed them, as well as schedule students for the labs. Okay. Instructors should also request and access material to assist students. For example, Sue Smith provided a introductory 20-minute video on how to use the lab. Nanslow also has written information on how to use the lab. And then the instructor, what I did was then this is a synchronous course, so I created a live synchronous lecture 1 week before the lab. So we went through the lab and the lab procedures. We talked about mitosis, what they're looking for in meiosis. And then a week after the lab, we had a follow-up lecture to discuss any questions and talk about expectations for a lab report and how to write a lab report. Okay? That information was already written in the Blackboard shell, but if we can use both auditory and visual, it seems to help the students more to hear it versus having them read the instructions. Okay, so we have both of those options now, which is fantastic. I also prepared, welcome Heather. I also prepared a photo gallery. So when we were in the test session, I took screenshots of all of the slides. Actually, I took several screenshots. And I put them into my Blackboard shell. And I told my students, I want you to concentrate on the lab itself. I want this to be a positive experience. Don't worry about the technology. We will take care of that. If for any reason you cannot capture an image, you will have a photo gallery present where you can pick up that image. Okay, it's in your Blackboard shell. And I think that was really important for my students to know that they had backup if they needed it. Okay, and I don't believe, and Albert can contest to this, that anybody used my photo gallery, but it was there. And then finally creating a grading rubric, right? The expectations of that final lab report. So here is an example of Sue Schmidt's video and introduction. It's wonderful. She uses this nice calming voice, which is the opposite of mine, so it's fantastic. And you can see that she has left you with websites to access, and certainly my email is on the top of this presentation as well, and I can send you the information that I have used and seems to help. We also had written instructions, again, provided by Nanslow. So our students really had a choice. And then the lab worksheet itself, now this is just a screenshot of the top of the lab worksheet. So this was the original lab worksheet. And remember, it had oh gosh, I'd say approximately, what would you say, Dan? Maybe 13 pages?
Yeah, something
maybe?
like that.
Would you say? Yeah, very, very comprehensive. And so I did take that for this particular class and modify it and simplify it. And then again, the picture gallery, which is in the Blackboard shell. So in case they couldn't capture one image or something happens, they can go into the picture gallery in Blackboard, pick out the onion root, count, for example, the cells, because they needed to count cells and see how long, for example, an onion root with mitosis, how many sections were in mitosis, how many sections were in interphase, and then the percentage of. Okay, so they had this information if they needed it. Now, equally important, however, is student preparation. And you know, we know that there's always one or two students that aren't going to prepare, and that really does not work for this lab. So they were required to review the written laboratory instructions ahead of time, attend the live synchronous lecture, and if they couldn't attend it, that lecture was recorded, review the video and written lab or written lab procedures, whichever they preferred, review the necessary instructional and reference material, and then I was really looking at and emphasized process versus perfection. It was important for the students to understand that the goal of this lab was not perfection. It was going through the process, trying new things, using the microscope, and then integrating that information into an objectively based lab report. Again, remember, most of these students have never written a lab report, right, or used objective medical terminology, yet they are in a medical coding program, right? So this is going to clinically relate over quite nicely. So I was really looking at it for clinically relevant assessments because the students are required to use, welcome Genji, because the students are required to use critical thinking skills during assessment, and assessments are in alignment, there's that quality matters word, with course instruction, objectives, and materials. They needed the ability to collect data, interpret that data, and create an objective lab report. Again, that required the students to analyze data and compose conclusions using the scientific method, which is not easy for non-science-based students. And so this relates over into their field. I felt that this would give them a better ability, A, to decipher a medical record, B, to read and interpret medical journals, and C, to read and write using an objective format. You can see down below in the text box that Sue just texted us and said that the full meiosis and mitosis lab activity is available, and she sent you that link. I would certainly recommend, if you don't have it, that you print that out. It is very impressive. Now let's talk about student outcomes. So what happened? All this planning, what happened? Well, the majority of students were able to produce a clear and concise laboratory report with the expected requirements. Now, there were one or two students, remember this is a small class, about 10 to 12 students, that were given specific feedback and allowed to make changes and turn their paper back in. Okay? Most of the students, though, the vast majority, required minimal feedback, and all changes were made successfully by all students. So I thought that that was pretty exciting. So we used a student survey. Again, very, very small population. So I'm just grouped together the positives, some things that went well, and then some things that we can work on, I can work on. Okay? So the majority of students agreed or strongly agreed that the remote lab was rigorous and challenging. In fact, everybody agreed to that, and it made them really think. The remote lab gave them the opportunity to work with other students. We had 4 students in the lab at a time collecting their data, and then they had the option, A, you can take that group, or you can take any group and write a group report, or B, you can write your report individually. It's your choice. They also said they feel like they had help from the instructor when I needed it, and everybody was so positive about the help that they got from the technical assistance. In this case, I know Albert was key in success for these students when I needed it. So if someone had a bad connection, if there was a problem with their computer, Albert would just reschedule them. So every student successfully went through the lab. Welcome, Lavona. And they also said the remote lab was convenient and easy to schedule. Some of the things that were fairly divided were that improvements could be used in the following areas. The remote lab had clear and easy to follow instructions. I believe that I myself could have simplified the introduction to a more appropriate level for these students. I also believe that in my live lecture, I could have given them, instead of schematics, actual microscopic slides so they can more easily identify mitosis. And then the remote lab was easy to use. Well, we didn't have the video introduction at that point. We had the video introduction after that point. Our slides were held up in customs because of the federal shutdown, so we weren't sure initially if we were going to be working through Denver or British Columbia. There are two different protocols there, so there's certainly some improvement that could be made. And so I thought about things that worked and things that I myself personally could do better. New video recording for the students, which is done. So Sue Schmidt did that wonderful video. Pre-laboratory quiz. We're always going to have students that are not going to plan. And so actually this was Susan Merkovich who was in the lab with me. She said, you know, I think a pre-laboratory quiz is a great idea. And so the idea would be they take the pre-laboratory quiz and they have to pass the pre-laboratory quiz. If not, they have to take it again. I would continue the live lecture, as I said, with more specifics on how to use the lab and review screenshots from the lab as well as actual microscopic slides on mitosis. I'd continue with the live lecture follow-up after the data collection has been completed. In particular, when asking for a student to provide a lab report, but I think that's important regardless. Continue with the photo gallery. It really takes the stress off the students who want this to be a positive experience they can enjoy. And provide feedback on lab reports, especially for non-science majors. So in conclusion, while some students felt they had difficulty with the lab, the majority of students reported that help was readily available. And the majority of students were able to analyze, compare, contrast, and integrate information in a successful manner. Those that were not needed minor changes in their lab reports. They were given feedback, and everyone turned in their second lab report. And so everybody, I would say, did quite well. Student instructor and preparedness is key to successful and positive lab experience. Okay, and I can't emphasize enough the link that you have between the director of the lab and the instructor. Albert Balboa, of the British Columbia lab director, was incredibly helpful. Again, made sure that all students successfully completed the lab. If they needed extra help, had computer difficulties, he was there to help them, and that really puts our students at ease. Think we can see the feedback for the full survey? Yes, you can. I need to. I did tally marks, Maria, but you can. I'm going to actually put it onto Qualtrics for next year. I just didn't have time this year, but yes. So no problem. So now we're going to move on to Dan and the lab. Dan, I wanted to just talk briefly about mitosis and what they're looking at. It'll probably take me about 3 or 4 minutes. Can I do that? Or would you like to
No, I think that would probably be very helpful. Just as a quick reminder, I've got a demo with PCC instructors or faculty right at noon, so we have to I have to use the same microscope with them. So I've asked them to delay that till about 12:10. So, but yeah, go ahead.
Okay, guys, this is going to be quick. We don't expect you to be masters of mitosis. Okay, we will help you with that. I just wanted to say that when performing this experiment, again, we don't expect you to be an expert in microbiology. Mitosis means cell division. For those of you that are not familiar with that, what happens is the chromosomes, our genetic material within the cell, basically duplicate. Uh-oh. Somebody is sharing the application, and I'm going to stop you.
Yeah, sorry about that. I didn't mean to click that yet.
Can you stop it on your end? Okay, thank you. Very good. Okay, so basically it means you have one original copy of the cell and one duplicate. All right? We use mitosis to replace damaged cells, repair cells within our body. So here's what you're going to be looking at. Mitosis includes prophase, metaphase, anaphase, and telophase. You're not going to be looking at prophase. I'm going to whip right through it. What we're going to be looking at in the onion root is metaphase. Okay? Metaphase schematically is where the chromosomes line up in the middle of the cell, okay, preparing to divide. So I want you to take a look at this right upper hand picture when you look at the microscope and see if you can identify this type of cell. Anaphase is easy to identify as well. You can see that the chromosomes, sister chromatids, are dividing, right? They're starting to make two cells. So you're going to see something like this. And then finally, telophase and cytokinesis is fairly similar. We'll call it cytokinesis. You see that we're creating two daughter cells here, and then the cytoplasm is going to split, and it's going to make two separate cells. But what you're looking for is that process of cytokinesis. Okay? And so you can take a look at these screenshots. Dan, it's all yours.
Okay, well, let me see if I can. All right, it doesn't give me the option to share the microscope. Sue, any advice there?
Would that be Sue Merkovich?
Sue Schmidt.
No, I think that would be Sue Schmidt. You know, you should be able to just go to the LabVIEW software as Albert had done in the past and just pop that up.
I've got that up and running, but when I go to share application, it doesn't show up in the list.
Oh, okay, wait. No, it's the sharing here is complicated. So you need to go, did you go through Tools?
No.
Go through Tools, go to Application Sharing, and then share your entire desktop.
Okay.
Okay, so I'm going to pop back to the whiteboard so you're off of that. Okay, so go up to Tools as Sue has suggested.
Share entire desktop?
That's how I have to do it.
Okay, I thought I could share a specific application, but okay.
I have had to share my entire desktop, then it lets me choose from the applications on my computer.
Yeah, for some reason it does not let me choose from any applications. I don't know. What are you guys seeing right now?
My lecture.
I just went back to the whiteboard so that you could go up to Tools and Application Sharing.
Okay, I did that, and I shared my whole desktop.
And then you should see a rectangle that just has things listed on your desktop.
There we go. Okay, so Dan, we see your desktop. Now select the LabVIEW software.
Yep, I did that.
It may take a second because we do see your entire desktop right now.
Okay. Yeah, it doesn't give me any.
Whoops.
Waiting for the web tour.
Okay, it shouldn't be a web tour because we're not doing a web tour.
When you went to Tools and shared your entire desktop, did you see like a rectangle that had a list of everything that was on your desktop?
No.
Oh.
All it says is share entire desktop, and that's it.
Okay, then go back to Tools, and then go to Application Sharing. Stop sharing what you're sharing right now, okay? And then go back to Tools, Application Sharing, and start sharing and see if that works. If not, someone else will have a suggestion.
Nope, it only gives me share desktop as the only option.
Oh, there's Susan. Susan, get on the mic, will you?
Time set. Hi, can you hear me, Sue?
Yeah.
Dan, make sure that your desktop, make sure that the lab is open, and then try to share the application. Because sometimes Collaborate won't list anything that's down in the tray. You kind of have to open it up first.
No, I know what it is. It doesn't list anything that's on a second monitor. It only lists what's on the primary monitor. So there we go.
Okay, good. I knew it was something like that. Thanks.
Okay. Sure. No, thank you. Yeah, I had the microscope window open on my big monitor because I thought it would be better, but it isn't. Okay, so now you should see.
Okay, and just to clarify for participants, because I just want everybody to understand this, that when we're in Blackboard Collaborate, we are simulating the experience you would have if you went right into the LabVIEW software. So Sue had mentioned earlier that the response is a little slower than if you were all in the LabVIEW software. But the only way we can allow you to see this, what's happening at the whole audience, is to do it through a Blackboard Collaborate share feature. So as we say, it has its little nuances. So don't attribute that to the LabVIEW software. This is not what happens in the LabVIEW software. This is what happens because we're using Blackboard Collaborate so that all of you can see what we're doing and share this application. Thanks.
Right. So the video in particular is going to look choppy on your screens because it's being shared to you through the Collaborate server, and that just kind of slows things down. So the students, when they actually use this, they don't see a choppy video screen. It looks very smooth to them. So just very quickly, looking at this control panel from the top left to the lower right, and this is basically the same design. We use the same basic layout that Albert came up with for the remote control panels for all of our experiments, and that is to have the equipment controls on the left half of the page and the video and video controls on the right half of the page. And so in this particular case, we have the controls for the microscope stage. You can move the stage back and forth, in and out. And you can control focus with the up and down. And you can switch that from coarse to fine, which just gives you a much smaller control, much smaller movements in the z-axis. There's also an automatic focus button, which on this particular microscope is of kind of limited application. It works best on the 40 and 60x objectives. But as you're going through, you can change the objectives from 4 at the lowest level, up to 10, and all the way up to 60. So, and each time you change it, it's best to focus because these particular microscopes are not very parfocal. So it tends to not quite be in focus when you switch to the next higher objective, for example. You can control the way the image is displayed under the microscope image box in the lower half of the left-hand side. And you see also there, I have instructions for how students can call into a voice conference so that they can talk to each other and collaborate, and also talk to the lab technicians who are monitoring that same voice conference. And on the right, these are the controls for the pan tilt zoom camera, which we're not viewing through right now, but we can if we click on picture-in-picture. We can see the view from an external camera that's sitting sort of in an operator position in front of the microscope. And we can see that the presets are set to show specific parts of the apparatus. You can see the pop-up list there, I hope, that comes up when you hover over the buttons. And so if you want to look at the objectives, you can do that. You can see that they change when you change them. You can look at the external camera controller, the objectives, or the oculars rather, and the condenser, etc. Okay, so that's a quick tour. Who wants to take control of the interface and do something?
Hey Dan, do you want to start a little bit farther on than we had planned, maybe starting at 20 magnification and then having them use the autofocus and capturing the image just in case you run out of time? Or are we going to be, we may not be able to go through all 7 steps?
I think we probably got enough time, but let's just see. Why don't we start on 20x and go from there? So do you want to go ahead and give some guidance?
Sure. So did we identify a volunteer? Be brave. It's easy.
I guess raise your hand with the little hand button if you want to do this. And then Sue Schmidt will tell you how to take control of the interface through Collaborate. No volunteers.
Great. So Kai, I'm sorry if I did not say that correctly. Let's start with you. Sue, you go ahead and tell her how to do that, and then I'll ask her what to do. How does that sound?
All right, that sounds good. The first thing you want to do is you want to go up to Tools, and you want to go to Application Sharing, and you want to request control of a shared application. When you do that, Dan will get a message saying that you want to take control, and as soon as he allows control, which just is a matter of seconds, then you will be able to move your cursor into this LabVIEW software and do functions.
Yep,
the one thing to remember for each person taking control is your cursor acts a little different than it would normally act, so it'll be a little bit slower when you take control of that. So go ahead first and request control, and Dan, let me know when
she's got it.
You have,
she's moving the mouse.
Okay, perfect. All right. So we had instructions on different activities that we were going to do. So Sue, do you have those in front of you for the first activity, what that was going to be? I think it was the lab presets, as I recall.
Yeah. So this is Sue B.
I know.
We're going to just start with number 1, and this is basically so everyone gets the idea how this works. Now Dan already showed us this, but Kai, if you could go to the Picture in Picture button, do you see that?
It would be on the right-hand lower side, and to be able to use the picture-in-picture function.
So click on that. Okay, and so again, that's going to show you the actual lab in Denver. You can select and show various lab views, and you can also zoom in and out. You see that pan, tilt, zoom camera controls? You can go ahead and play with that, and then you can zoom in and out. And again, it does not lag like this in the real lab. And I think Albert has commented that the students like to use this in particular to take a whole look around the lab. Is that right, Albert?
Yeah, that's the first thing almost every student does is checks out the environment.
You get to see how messy our lab is right now. That's our server case with all the computing hardware in it. That's DJ's desk.
Also, as you're scanning, please keep in mind this is only one lab activity that NAMSLA is doing. I think a lot of you already know that. As far as biology, I know they're working on hematology, urinalysis, disease cells versus healthy cells. Many other experiments are going on. When you're done playing, and feel free to take your time, we'll move on to the next topic. But go ahead and explore all you want. Okay, great. So we need a volunteer for number 2. You're going to be moving the stage of the microscope, and I'm going to tell you what you're looking at in just a minute. So raise your hand.
Okay, and logistic-wise, sorry, Dan, you need to release her. And then the next person will also have to go up to Tools that has volunteered, Tools, Application Sharing, and Request Control.
And that's Lee.
Okay, I think Kai still is moving my mouse. Hold on. There we go. So, all right, I think I took control back away. There we go. And you have it now, Lee. Oh yeah, control space. Okay,
so Lee, what you're all looking at in the right side of the screen may not look quite familiar yet is the onion root tip. And we're going to see later the stages of mitosis. But what we'd like you to do, Lee, is go up. We're actually right where your arrow is at stage controls. You're already there, aren't you? And go ahead and move the stage of the microscope, and just with the view you have, try to get that onion root positioned where there are a maximum amount of cells. Put it right in the middle of the screen, that area. And then when you're happy with what you've done, if you think you've got it down, go ahead and let us know that you're done and we'll move on. It keeps looking better and better, actually.
Well, can you hear me?
Yes.
Okay. Okay. I'm not a microbiologist, so I'm not quite sure. Is this looking fine here?
Yeah, I think you did good.
Okay.
Okay. So thank you, Lee. So we need a third volunteer to raise their hand. Now we're going to get into magnification, which is fun. So, third volunteer.
And you know what happens when your students don't volunteer? We select you.
I know, this is terrible. I don't want to put anybody on the spot. All right,
there we go.
Great volunteer, Angela. Thank you, Angela. You have your away on, by the way, so we think you're not in the room. So if you want to click that off. All right, so give Angela control, please.
Yeah, go ahead and request, and I'll approve it. So I think that's what, tools?
Yeah, Sue, could you remind each time? It gets somewhat confusing.
Yes, I can do that. So for the Angela to take control, it would be tools, application sharing, and then request control of shared application.
All right, she's got it.
Great. Okay, Angela, what you're going to do now that Lee did such a nice job with the placement of the onion root is you're going to go to the objective selection. You're going to use the 10-time magnification. Okay, so you need to move that magnification from 20 times to 10 times. On the right, go right. There you go. Okay, so go to the other arrow. There you go. I did the same thing actually when I started. You might have a lag. Okay, one more time. Great. So you went from 20 to 10. Now we're going to start small and go big. It gets really exciting in the end, okay? So wait for that. Go ahead and hit the autofocus and let's see what we have. As Dan said, it may not work as well on this one, but go ahead and do it anyway. And we are all having to wait for it. Again, you don't have to do this in lab to autofocus.
Yeah, normally when the students are looking at the same interface, they're not sharing it like this. They're each looking at the same interface simultaneously, and it's an immediate thing. So when she's clicking automatic focus, I see it change immediately.
Right, and now I'm not real. I mean, I saw it clear, then I didn't see it clear. Let's do this because we're going to move up from 10 anyway. But what I want you to do you think you can go, Dan? Do you think it would help if she did the up and down controls on the 10 times to make it clearer?
Yeah. I think if you go down just a couple of clicks, you'll probably be pretty close. So the up and down in the stage controls box up there?
Just to the right of that.
Yeah,
not the stage control itself. To the there you go. Click
there.
Yeah, down a couple times.
Yep, that looks pretty good.
There we go. Okay. Now, gets exciting. What we want you to do is go down the screen. You're going to see where it says Capture Image. Just straight down where it says Capture Image. Click on it. Now go down to where it says View Captured Image, just to the left and down a little bit.
Yep.
Yep. And then this is going to pop up on everybody's screen. Now, again, there's going to be a lag that we don't usually have. Okay? And let's not capture an image, Dan, until we get to the really good one, okay?
Sure.
Okay. So what you see is the onion root tip captured image, but really we can see that there are a lot of cells in there, but we really can't tell what they're doing. So let's go ahead and get rid of that box. Yep, delete it. And let's move on to the next brave volunteer. And you're going to go to 20 times. We're going to go up to 60. So we're going to do 20 times next and see the difference. And we will get to see some of the cells that way. Who's next?
And again, just to let the person who volunteers know, you go up to Tools, Application Sharing, and Request Control of Shared Application. And Dan has taken control away of the previous volunteer. So we're waiting for that next volunteer so we don't have to pick on you.
Maria, see, I saw you volunteer earlier, so great. Maria, take it.
All right, you've got it, Maria.
Okay, so Maria, you're going to do exactly what the person earlier did, except you're going to go to 20 times the magnification. And then, Dan, she should be able to use autofocus fairly successfully.
Yeah, probably.
Yeah, but you're still getting new microscopes, right?
We've got them. We're just working out some control issues with them, but we've got them.
Okay. So take that objective, Maria, and click it 20 times to go to the right arrow.
Well, only click it once.
Nice. Now, and then you're going to try the autofocus, and let's see how that looks. To the left, under the digital microscope. That looks pretty good. Okay, so let's go ahead and, if that sounds good, Dan, we'll capture the image.
Sure.
So Maria, go down, capture the image, and then let's all take a look at it. So then click on View Captured Image. Wait, you didn't capture it. There you go.
Yeah, she did.
Oh, she did?
Yep.
It didn't light up. There we go. Okay. Oh, I see. Okay, so we are looking at an even closer view of the onion root tip, and still pretty hard to see exactly what's going on there in regards to mitosis. Beautiful picture, however. So let's move on. Maria, if you want to delete that and get it to 40 times.
Yeah.
Then we'll be able to see what we're looking for.
Yeah. So remember on that picture that you can also click on it to zoom in quite a bit too.
Oh, that's right.
And you probably want to go ahead and hit autofocus there too, but it's pretty close. And you can try using the up and down buttons if it doesn't look fully focused, but it's pretty good.
And even here we can see the differences in the cells. You want to capture the image and view the captured image?
Oh yeah.
And as Albert suggested, talk her through also magnifying what is already being viewed.
Just click anywhere on the picture. And now you can click and drag it, although that might be kind of difficult because of the lag, but you can move around that image. Oh, maybe not. You might have to use the slider bars actually on the window.
And it's a little bit fuzzy on our end. It's not quite as clear as the unmagnified picture.
And just to let everybody, know, the students at this point can each view the captured image simultaneously and independently of one another, and they can also save it to their local computer. In this case, if you saved it, you'd be saving it to my computer because that's where it is, but the students can save it to their own computer and then later use that in their lab reports.
So do we want to demonstrate
capturing image? Go ahead.
Yeah. Do we want to demonstrate that now or go up to the next magnification?
I think we should. Could you go ahead and get out of this window for a minute and go back to 40 times, please? I think I want to do it at 60 times, but could you do this captured image one more time, please? Oh. Without the magnification? At 40 times?
You just want to view the same image again?
I do.
Okay.
Come on, that's the biology geek in me. I just wanted to talk a little bit about how you can start to see the cells. Yeah. So I don't have a pointer here, but you can clearly see, can't you, that some of these cells, you can see little strands. You guys see those different cells? You can also, oh, thank you very much. Very good. Thank you. Now go to the one just below that, the cell just below that with the 2 dots. So that there is the process of cytokinesis, where the 2 daughter cells is separated, and now they're getting ready to separate into 2 different cells. Everybody can see that?
Mm-hmm.
Okay, sure. Let's go to 60 times. Do we have a volunteer?
We've got about 5 minutes left.
Okay. How about one more volunteer?
Sorry, 8 minutes.
And then we'll go 60 times and capture the image.
Okay.
So again, Dan has released Maria's control of this application. And so the next person who volunteers would go up to Tools, Application, Sharing, and Request. Thank you.
And that would be Christy. So Christy, go ahead and do that. And I also want to let you guys know we're looking at things digitally, right? And so that's how we look at MRIs. That's how we look at bone scans. That's how we look at CT scans. Everything is now digital. And so I also think that this makes, again, that clinically relevant connection because we are now a digital world, in particular in the medical field. Okay, does she have it?
Yes.
Okay, click to 60 times, please.
And using the 60 times objective means you're looking at about a 600 times magnification, if I recall correctly.
You know, I can't remember if we used 40 or 60 times that was the best picture. That's pretty nice right there, I have to say. And you did autofocus. Go ahead and let's capture that image and view the captured image, and then Dan will talk you through everything else. Let's take a look at the cells first.
So you can see that the captured image is a lot higher quality than the streaming image that you're watching on the control panel. And so we asked the students to use these captured images to do all their analysis.
So you can see how nice and clear this is. And one example of the lab is they need to take 50 cells in a dense area like Lee found for us, and they need to count them. They need to identify the phases that they're in, and then the percentage of time that they're in those phases. Okay, do you want to talk about captured images? Or
sure.
How to do it?
Here's how the students would capture it. If you right-click anywhere on that image, yep, and then you can, a couple different ways, but you can copy if you go down a little farther. You could print it also, but if you copy it, then you could go and now you could paste it into, say, a Word document, for example. So that's all there is to saving it to your local machine. it would be difficult to demonstrate that right now. I'd have to open Word and that sort of thing. But basically, that's what you'd be doing. Does anybody have any questions about that? No? Okay. What else do we want to do, Sue?
Well, the last thing we were going to do were the color options, but if we don't have time for that, that's okay. I think that we really went through the important items, displaying from, small magnification to greater magnification and the possibilities and how clearly we can see, and a better visual field than we could get, for example, with our undergraduate microscopes. So.
Yeah, I don't have a printer that's accessible on my computer, so.
What I ended up doing initially is using Snagit and getting them the pictures that way. So there are many ways that you can do it.
Yes. The downside of using Snagit or other screen capture software is that you're limited to the resolution of the actual screen it's displayed on. If you save the picture or copy it and paste it, then you get the full resolution. But you can do it either way.
So if you just use the save feature and just direct that image to a place within your computer, say picture as. So you won't see the rest of this right now, but in this case you're in Dan's computer, and if you were to save it, this image high quality resolution would be saved to his computer. Let's go ahead and back off of the image, because we do want to point out just the color differentiations, I think, Sue.
Sure.
So maybe Christy can just quickly go through a couple of those. And of course, these different contrasts that you can use help if you are trying to get a different quality for your image for your lab report. So there may be a different color combination that works better to actually display what you are representing in your lab report. So it does offer you these options, and when you do capture the image after you have applied these colors, you will get the image like if it were a negative or black and white, etc.
We're live on here, and I'm on the conference call waiting for playback.
Albert, do you have anything to add as the other resident professional with the labs?
Okay, I thought that the way that we're doing this demonstration right now could be used also to prepare students to actually do the lab. So you could use Collaborate or Adobe Connect or whatever you're using to explain about the lab without the students having to log in. They could even take control of the application the way the participants are here. Also, you could get it to use this for people at a larger crowd rather than just 4 or 5 students at one time in there.
Yeah, that's a good point, because for actual collaboration between students, we usually limit that to 4 or 5 students because it's a good-sized group, and also when you get too many people on one directly accessing one machine like that, it can start to lag. So in order to make sure everybody gets the optimal experience, we limit the group size. But in a situation like this, even though the video is a little laggy, but we can share it with a lot more people. All right, so we probably need to go ahead and wrap it up because I've got to move on to the PCC faculty.
Yep, I would just like to open it up very quickly to any additional comments or reflections that participants in today's demo might have in relationship to viewing this demonstration. All right. So with that, it is noon and we will be closing the session now. If you do have any additional questions, observations, comments, we would love to hear them. you feel free to communicate with Sue Buie or with Dan Branan or with Albert. Could each of you input your email address in the chat window so people know how to reach you? And also I will put my email address in the chat window as well, because I'll be happy as well to receive information and communicate it out to people in relationship to what your questions are. So we'll quickly do that so that everybody has that information. I want to thank all of you for participating today and hope that this demonstration helped even to further allow you to see how this can be applied in your own classroom. So with that, I'm going to stop the recording and also just mention to everyone